Bacterial Growth on Raw Chicken versus Eggshells by Jernigan. Eggshells and raw chicken are contaminated with disease-causing microorganisms. That's why we're told to cook our chicken and eggs before we ingest them. A Consumer Reports study highlighted by the Center for Disease Research and Policy tested 316 different samples of raw chicken. On those samples, 43% of them contained Campylobacter, while 10.8% contained Salmonella, both disease-causing microorganisms. The same study also found an Enterococcus species and Escherichia coli growing on them. And both these bacteria are normal parts of the human gut bacterial population. This indicates fecal contamination. Almost half the samples, 49.7%, tested positive for at least one multi-drug resistant bacteria. This means the bacteria was resistant to 3 or more different antibiotics. Now when it comes to eggs, according to the Center of Disease Control, poultry can carry bacteria like Salmonella, and they can contaminate the inside of the egg before the shell's even formed. And the eggs can also become contaminated from the droppings of the poultry and the environment that they're in. So the raw chicken sample will grow more bacterial colonies than the eggshell sample because raw chicken encounters more organisms during the packaging processing unit than eggshells do. When chicken's being ready to be sold in stores, they use knives and machines to prepare it, and they don't decontaminate these, which can spread disease-causing microorganisms. The experiment did not test the type of microorganisms growing, just how many bacterial colonies were produced. So it's not clear if the bacteria was disease-causing or not. For the experiment, raw chicken, eggs, 2 nutrient agar plates, 2 cotton swabs, a ruler, and sterile water were used. First, we had to measure 5 centimeters on the chicken and egg. Then we had to dip the cotton swab in sterile water and swab the measure 5 centimeters on the chicken and egg. Then we dragged the chicken sample once down the middle of the nutrient agar plate and repeated for the egg sample. We then used a toothpick to go back and forth on the agar plate to spread the samples, and then let it grow for 24 hours. After the 24 hours, we counted how many colonies grew, and these are the exact numbers for each trial. So we then calculated the average number of the colonies grown in the 24-hour period. The chicken grew an average of 181 colonies, while the egg only grew 81.25. So the experiment supported the hypothesis because more bacterial colonies grew on the raw chicken sample than the eggshell sample. And the results may have been skewed by the contaminated supplies, like the ruler. It could have already had microorganisms, and so then when we measured it, those microorganisms could have then been transferred to the sample. And we only conducted 4 trials. So for it to be a more accurate experiment, we would need to do more trials. Now, the chicken swab produced an average of 99.75 more colonies than the egg, showing that the chicken does contain many more microorganisms. And as stated earlier, chicken can cause disease, and occurs when it's cross-contaminated with other things. For example, if you cut a chicken on a cutting board and then cut a carrot on the cutting board, that carrot is now contaminated with a disease-causing organism from the chicken. So a study was conducted by the Environmental Health Specialist Network of the Center for Disease Control and Prevention. And the study stated that the chicken was the 4th leading cause to foodborne illness. It also found that 61% of the foodborne illnesses came from restaurants and delicatessen. So they decided to interview managers of restaurants to see how they cook their chicken. So 54% of the managers reported using other methods to determine the final temperature of a chicken instead of using a thermometer. Some of these methods included the chicken's appearance, which occurred in 47%, chicken's feel or touch, which was 28%, they used a timer, 29%, and experience and skill, 46%. Over half the managers said that they rinsed or washed the chicken. And this process may reduce the pathogens, but it can also create cross-contamination because while washing, the water can splash and get on other surfaces and around the environment. In the end, one must be sure to prevent cross-contamination so that others don't get sick.